Hello and welcome everyone to this podcast on DNA and chromosomes. Now we're only going to spend one day on DNA and chromosomes. Some of you have already taken a genetics class, some will take one in the future. But there is this rumor on the street that some of you won't ever take a genetics class. And so it's important we talk about it a little bit. And I want to begin by showing the central dogma to explain why it's important to understand the DNA, its structure, how it's packaged into chromosomes, so it can be used. So DNA is transcribed into RNA. It can be transcribed into messenger RNA, tRNA, ribosomal RNA, microRNA, a whole litany of RNA molecules. When we talk about this process, we often refer to messenger RNA, but it's important to remember that transcription occurs to make all the types of RNA. That messenger RNA is then translated into protein. And we just spent the past several classes talking about proteins. We'll continue to talk about proteins throughout the semester, but in more of an applied fashion. What are the proteins doing in the cell? We won't spend too much time talking about RNA. I should add something here with the RNA, just to make it legal here. Before it's translated, particularly when we're talking about eukaryotic cells, it is processed. It has to go through some rearrangement and modifications before it can be translated into proteins. But we don't spend too much time talking about DNA. So we're going to talk about DNA in this podcast and probably the next one. It'll probably take 2 podcasts to get this all done. So why is it important that we spend some time talking about DNA? Well, first, in most organisms, it's the unit of heredity. This is what gets passed on to our progeny and then to their progeny and so forth. So understanding how it's constructed is important in understanding this process. It's also important when we think about heredity when we come to the topics of mitosis and meiosis this semester. We'll want to understand the chromosome quite a bit and how it's used to get packaged into progeny cells. The second reason we want to understand DNA and spend some time with it is because it codes for life. Not to be too dramatic, but the DNA sequence here ultimately decides which RNA molecules what they will look like and how they will be translated into proteins. And these proteins, by and large, determine our traits. Now, we know some RNA molecules never become proteins and they themselves play a role in determining our trait, but ultimately, that is all in our DNA. The third reason is that it explains our similarities and our variations. Okay, so now what I'm going to do is I want to step back and break down DNA to its smaller components, and then we're going to build it back up. Okay, so let's go ahead and get started. If we were to draw a double-stranded helix here, each strand here is made up of repeating units of the DNA's functional unit, all the way down here, and on both strands. And the functional units are nucleotides. Now, what are nucleotides? And we've already described these before, but it bears repeating again. Nucleotides, in this case we're going to talk about DNA specifically and not RNA. So the nucleotide for DNA contains this sugar, and this sugar is deoxyribose. And the reason we call this deoxyribose is because of this 2' carbon, it has a hydrogen. Ribose, if we get rid of the deoxy part, the sugar ribose found in RNA has an OH there. Now, off the 1' carbon here, we have a base. And we'll come back to bases in a moment. But in DNA, for now, what I want to tell you is that the bases can be adenine, thymine, guanine, or cytosine. For simplicity, we often just refer to them, as we're talking about them, as ATCG, or GC in this case. And then coming off this 5' carbon, we have a phosphate group. These 3 components, phosphate, sugar, deoxyribose, and one of these bases make up the nucleotide. Now, you won't need to know the structure of the bases, but there are a few things about the bases I do want you to know. I want you to know that they are either purines or pyrimidines. Purines contain 2 rings. You don't need to draw them and know the actual position of all the atoms in it. But what I do want you to know is if I ask you to draw, say, a purine, or be able to recognize a purine, look for the two bases, and if you have to draw it, I would draw it like this. Just so I know that you know that purines have two rings. Pyrimidines have one ring. And so if you ever have to draw it in association here with a nucleotide, I would just draw it as one ring. I put a square there. You could do circles, just so I get the point that you know one ring versus two rings. If you have a hard time remembering this, the way I've always remembered this is that the word purine is smaller than pyrimidines, yet purines have two rings, so it's kind of counterintuitive. The shorter word has more rings, the longer word has fewer rings. It works for me. Purines include the guanines and the adenines, the G's and the A's. The pyrimidines include cytosines. I'm going to leave this blank here for a moment. I'm going to add something there. And thymines. These are in DNA. And I'm going to tell you this third one, and this is only found in RNA, and it's uracil. So these three are the pyrimidines. You should know all three of them. And the reason why I always put them in this order is because it helps me remember that CUT are pyrimidines. And this sounds a bit corny, but when I was a student, I memorized things in weird ways. And I always think of a pyrimidine, I think of a pyramid. The top of a pyramid looks sharp. And so you could imagine you could cut yourself with it. So that's how I've always remembered that cytosines, uracils, and thymines are pyrimidines, because the top of a pyramid could cut your finger. That's a little embarrassing to admit, but that's how things roll in my mind. And while we're talking about bases, there's one more thing I want to mention. We'll come back to this a little later probably, but let's go ahead and look at it now. And that has to do with the fact that this base here and this base here, for instance, they form hydrogen bonds with each other. All of the bases form hydrogen bonds with each other with a nucleotide on the opposite strand. And they only do that in certain ways. And in DNA, that way is G's bind to C's, one purine, one pyrimidine, and A's bind to T's. Again, a purine and a pyrimidine. This is the only way they bind in these hydrogen bonds between the bases. I always remember that G's bind with C's because they both have a curve in their letter. And the A and the T bind with each other because they're similar in that they are made up of just straight lines. So I've always remembered that's how they pair. Most people can just remember it. I had to come up with a system. You may notice that when I did the G to the C, I put 3 bonds here, 3 lines, and I put 2 here. That wasn't a mistake. That's because when a guanine pairs with a cytosine in DNA, say over here, it forms 3 hydrogen bonds. Over here, adenines and thymines, they form 2 hydrogen bonds. A couple of things to keep in mind about that is that the GC interactions here are stronger. It takes more energy to disrupt them because there's 3 hydrogen bonds versus the 2 hydrogen bonds of AT. So they would be weaker. So if I were to ask you a question, say on a quiz or a test, or just as you're walking down the hall, and I would say there are 20% guanines in a segment of DNA. How many thymines are there? Or what's the percent of thymines? Well, you can figure this out because for every G there is, we know there's going to be an equal amount of C's. So there will be 20% C's. Together, these add up to 40%. So that means the T's and the A's must add up to 60%. And since there needs to be equal amounts of T's and A's, then there needs to be 30% A's and 30% T's. Okay, now I want to move on and look at the single and the double strands of DNA and how they build upon each other, taking each of these bases. The first thing I want to do is just draw one generic base. So I'm going to draw an adenine here. I'm showing that adenine is a purine by showing 2 rings, and then I'm going to put my phosphate here and the hydroxyl group there. This is the 5' carbon, so we always call this the 5' end. And we always call this the 3' end. Now let's add a second base. And though it matters that A pairs with T over here, and we'll show that in a moment, what comes next, there's no rule for that, except that ultimately that's going to determine a sequence of a gene perhaps. But any base in principle could come next. So we'll put a cytosine here. Again, I'm going to show that with one ring because it's a pyrimidine. We have our phosphate here, and what it will do is it will form a bond with this hydroxyl group. And to remind you from way back when, earlier in the semester, This bond that forms is called a phosphodiester bond. It's a special covalent bond right there. And you have your hydroxyl group here. The next one, let's make it a thymine. Thymines are also pyrimidines, so they're going to be one ring. We have our phosphate here, and it will form that phosphodiester bond with the preceding nucleotide. And let's go ahead and draw a fourth base, just so we can include them all, so nobody feels left out. And so this will be a guanine. Guanines are purines, so they have two rings. Also has the phosphate group, and it's going to form that phosphodiester bond with the hydroxyl group there. This will always end, the last nucleotide of any sequence will have a hydroxyl group there, and we call it 3' hydroxyl. So we always write DNA on one strand, 5' to 3'. Anytime you see a piece of DNA, if it's just a single strand of it, by convention, we write it 5' to 3'. However, it's the reverse on the other side. On the other side, it will be 5' to 3', and that's because DNA is anti-parallel, and I'll write that word down here. Remember, adenines need to bind with thymines, a purine to a pyrimidine always. And we should put two bonds in between there, so I know that you know there are two bonds, hydrogen bonds. Alright, so the next one here has to be a guanine, because C's bind with G's and they form 3 hydrogen bonds. The next one down here will be an adenine, also a purine, binding to this pyrimidine. 2 rings and then 2 hydrogen bonds. Guanines pair with cytosines, the single ring pyrimidine. And let me draw the sugars here. So this cytosine here, again, will at the very bottom have the 5' phosphate, 5' here. It will have the sugar like so attached to the base. We'll have our hydroxyl group here, which will bind to the phosphate group of this thymine. And this is going to form a phosphodiester bond with the guanine nucleotide here. And this guanine is going to form a phosphodiester bond with this thymine. And it's going to end with this 3' hydroxyl group. Again, it's anti-parallel. This side is 5' to 3', and this side is 5' to 3'. That's what we mean when we say anti-parallel. I know it's an ever so small error here that I want to point out. I'm not going to redraw this, but here I put phosphorus with 4 oxygens and another oxygen. There's really just a total of 4 oxygens here. So this really should be 3 here plus that one. So I'll correct that real quick in case somebody's keeping score. Now the ends was about 4. One thing you will notice is that a feature of the double helix is that the phosphate groups, the groups that carry a negative charge, they are on the outside. And so if you look at a double helix, the phosphate groups are positioned on the outside of the double helix. Something that was proven by one of my favorite scientists, Rosalind Franklin. And on the inside, it's protected against water. It's more of a hydrophobic environment on the inside. That's all I want to show you with the molecular interactions of the bases here, of the nucleotides. Next, I want to talk just a little bit about replication. Not nearly as much as you'd get in genetics, but certainly I want to bring out some features of the double helix and how that explains how information is passed on in a faithful manner. So if we have our double helix here with our correct 5' to 3' orientations, we know that in order for this to work at all and to code for the traits of life, that this DNA molecule has to get passed on to progeny, either through meiosis as we make gametes, or just as any cell divides to make a copy of it during regular cell division. So how does it do that? Well, there's a lot of different theories, and we're not going to talk about all the theories in this class. I'm just going to explain to you the way we know that it actually happens. So first, during replication, what happens is these strands are pulled apart. This strand and this strand are slowly pulled apart. And I'm going to draw it this way, but I want you to know that it doesn't really pull them all the way apart and then replicate it, but it's just easier to show it this way for now. So we pull apart these 2 parent strands. So we call these parental strands. So the red that I draw will be parental strands, the original strands here. Then it's copied, and I'll draw the new strands in blue. It uses the parental strand as a template to create the new strand with both copies. So the blue are daughter strands. Then as cell division continues, and we'll go through mitosis later in the semester, so I won't talk about it now, but this new DNA gets packaged up into a new cell. I should draw a cell around this one too. By this process, this DNA in these new cells will have an exact copy of what was in the original cell. We call this semi-conservative replication. And the reason we call it semi-conservative replication is the word semi means half. And so in each new cell, half of the original DNA from the original cell here is conserved. Half of the original DNA in each new cell is conserved from the original cell. The other half is new DNA. But since I used this as the template, it will be identical to the one over here. And though I didn't draw it that way, this of course would still form this double helix, as with this one. And we would have the correct base pairs and the correct positioning of the phosphates. The last thing I want to talk about in this first podcast on DNA structure are these terms minor and major grooves and why they're important. They form because of the way the double helix is positioned and the way that the bases are arranged. You don't need to remember 3.4 angstroms and 36 angstroms, but it's important to know that the way the DNA is shaped is based on physical constraints of the molecule itself. So as the DNA forms this double helix, this large gap here in this region right here is called the major groove, and this smaller gap here is called the minor groove. Over here, if we look at this bead structure here, you can see a major groove here where you see more of the lighter beads, and the minor, major, minor, major, minor, and then major. They always alternate. So why are they important? They're important because various proteins, be they transcription factors, histone proteins, or other DNA binding proteins, they will often have two features to them. They will recognize a specific sequence of bases along here, but they will also often fit nicely within the major groove or within the minor groove. These spaces here, these two grooves, often offer a nice packaging system where the proteins can wedge in here to recognize a specific sequence of bases. Okay, that's all I have for this first podcast on DNA and the chromosome. In this podcast, we talked about nucleotides, how nucleotides were structured, the parts of the nucleotide that is, how nucleotides were positioned along a single strand of DNA, how they paired with nucleotides in different strands of DNA to form the double helix. We talked about the anti-parallel nature of DNA, and we talked about how DNA was replicated in a semi-conservative mechanism. In the next podcast, we're going to talk about how this double helical strand of DNA is packaged into a very discrete unit. So if you have any questions over this first podcast, let me know. If not, I'll see you in class. Bye.